Hello students, welcome to my channel. In this class, we will see, first we discuss what we already done. PCR based site-directed mutagenesis is going on. So this was done. This and this will be covered in upcoming class. This is done. Now we do this and this. This is part of this one. Why I don't know why I put here. This is part of this topic. Okay, let's start with Megaprimer PCR method. Megaprimer. Although primer are usually short, as we see so many classes in which we study or learn about what is primers and how it is used in PCR, and they are short segment of the single-strand DNA, single-strand oligonucleotides. Long double-strand DNA can also serve as primer. This is the twist in the story. Large primer, this large double-strand DNA is megaprimer. So here two terms. One is primer and second one is megaprimer. Okay. Primer are short single strand DNA, and short means oligonucleotide are 15 to 25 nucleotide in number. In mega, there is long, means 80 to 100 nucleotide should be there, and double strand DNA is considered megaprimer. So we'll see how the megaprimer are used with the PCR. You can see PCR. Okay. Then megaprimer introduce. Why we use? To introduce point mutation, deletion and insertion. And what is the principle? Use 3 oligonucleotide primer. There is 3 oligonucleotide primers are used. Means 3 primer will be used. How? We'll see. 2 primer flanking primers and one is middle mutagenic primer. Means this is the DNA, and how we make from this DNA to 3 primer. 1, 1, 1. 1 and 2, these are flanking primers. And this one is the middle primer, which is mutagenic, in which we create some mutations, some changes. So, involve 2 rounds of PCR on the template DNA. For this creation, involve 2 rounds of PCR. We conduct 2 rounds of PCR to generate or use these kind of primers. How? We'll see. So, in first round PCR, this is a first PCR or first round of PCR. 1 outside flanking primer and 1 middle mutagenic primer are used here. We can take this one or this one, not and. Okay. So we'll take this one only. So one is flanking, first one flanking, and that middle one will use as star and then star. These 2 are used in first round of PCR. Then it produces a double strand fragment containing desired mutation. This product is called megaprimer. So how we create megaprimer? We take 1A outside flank, 1A middle mutagenic primer part, then we create megaprimer. How? We'll see. See, here is a DNA strand. and we are taking this middle and the flanking one. This one is flanking one and this one is the middle where we done our mutation or mutation one. Here we create some mutated or add different nucleotide which are not complementary with this sequence, but this flanking will be attached to a complementary, and then we make or just force to do bonding between these unmatching area. So yeah, so this is how we make our Megaprimer, by producing double strand fragment containing desirable mutation. Here is we have desirable mutation and this is the double strand. This is how we create Megaprimer. Then in second round of PCR, what we do, we'll see. Megaprimer is hybridized to wild type template DNA. PCR is carried out using Megaprimer and other flanking primer. Now, in this Megaprimer, we make Megaprimer. This is Megaprimer. Okay. And then we make to hybridize this double strand to get hybridized with the wild type of the DNA template. When they are carrying, and this is carrying what? A Megaprimer and a outer flanking region. This is one flanking region. This flanking, because we just add here our primer only. We don't make this remaining part. We don't synthesize that remaining part. That will act as a now flanker. This is the second flanker here. And this open part will complement with the wild or template DNA, single strand DNA, wild type, that will attach in this area. And then generate a fragment of same length as original target DNA containing desirable mutation. Now we make this equal to the wild type of DNA template. So now we get large size or same length to the same to what? To the wild type. So now we make our desirable mutated DNA, large DNA part which is similar to the wild type except from our desirable places. So what is the key feature of this? The amplified product from the first PCR is act as primer in second. As I make you to understand, this one is used as a primer for the second PCR round. So final result is entire DNA molecule contain desirable mutation. Easy, okay. So Mega PCR, so the Megaprimer PCR is done. Now inverse PCR method. What is inverse PCR method?